Hello. How's the audio? Maybe you guys can wave or give a thumbs up. Yeah, thanks. Thanks very much. Great to see so many people popping in here. Right on time. Got a great crew. Hello, hello. Let's see, am I spotlighted at the moment? Okay, I'm gonna turn off the blur for a second and then I'll just show you what we've got going on in our room here. There's our gang. Hello, hello. Okay, that's an interesting view. I'm seeing it in real time on our giant screen. Look at that. Whoa, give you a headache there. Okay, but that's our conference room that we're in. Hello, people from all over. It's great to see you, and we're so appreciative of your support and energy. We got our advancement philanthropy team here, and our new and our exiting clinical fellows, and our Merkel collaborative team, leadership team. Hello, it's really wonderful to see you. And folks are still popping in here, but I will go ahead and start showing you a couple slides. We're going to keep things pretty informal. We want to definitely leave plenty of time for your questions. The thought will be to put questions in the chat, and you can do that anytime you want to. But we will be recording this. I think you probably already got your little irritating pop-up thing that says the meeting's being recorded, so you can get rid of that if you press the button. But this will be shared with folks who couldn't make it in real time today. So yeah, we're really happy to see you. I think my name is Paul Nghiem, and I lead this Merkel program here at University of Washington and at Fred Hutch. There is a lot going on. A lot of it's really, really, really good, and a little bit, especially coming from our NIH sources, is not what we were hoping for at this moment. I'll give you a little bit more information on that, but we'll get through it. And we're really happy to give you an update on where Merkel research is going. And then one of our big highlights of the day is that Geraldo Rodriguez, our senior graduate student who is completing his PhD in a couple of months, will tell you a little bit of an update on some interesting science and how your gut microbiome is linked to your immune system, and we think probably how we're going to beat Merkel. How many people do beat it and how we can make it even more likely. So I'll start with where we're coming from. I think you all know that a few years ago, if a person developed advanced Merkel cell carcinoma, their chance was only about 5% of having a good outcome by a few years later. If it advanced and if you were using chemotherapy, which was the best thing we had at the time. At that time, we were getting a small amount of money in philanthropic dollars. And
Speaking this way. Now I can't hear him.
Yeah, no, hold on.
Oh, you cut out.
I think it's on mute.
something. Oh, it keeps auto muting.
Yeah, there you go.
Does somebody else have control of the room? No. Okay. Okay. Sorry about that. So hopefully you got the concept that we were only about 5% success with advanced Merkel a number of years ago, and that went up by tenfold to 50% based on immunotherapy starting around a decade ago, almost. Less than a decade ago, we then had multiple approvals. So that's really transformed care for patients. And our team does a lot of things, including studying the basic biology of this cancer that led to those improvements and conducting translational research that helps to transform discoveries into clinical interventions for patients, which has been so gratifying to watch over and over these past years. And okay, we need to be able to mute people. Jim Seaman. So everybody can watch. Maybe you can make more people co-host and they can watch. Thank you. Yeah. And then we're able to really changed the way that this cancer is managed in so many ways, including most recently circulating tumor DNA, which is just transforming our ability to see this cancer come back early and know so much more about it. Then in January of this year, we launched the Merkel Cell Carcinoma Collaborative Institute that is changing the way that we work with people all around the world and is already a very exciting, important piece of progress. So I want to touch briefly on philanthropy. It's important at many levels. And before that, I'll just say the one sentence that after years of working on our big $15 million grant and writing the 700-page proposal twice, and being told in December that we had a top rating from the National Cancer Institute, the decision for funding it in February was postponed until June 10th. June 10th was Tuesday. They had that meeting, and at that meeting they said, we have no idea if we're going to be able to fund this or, frankly, much of anything else. The fact is, the current budget in front of Congress calls for a 37% totally unprecedented cut in NIH. If that goes through, all of the current grants that are funded will have to be cut or eliminated because there isn't enough money to pay for current grants. So the concept of a new grant like ours that just happened to be coming up for renewal at that time, getting funding, is something I can't imagine how hard it is to be in the NIH at the moment trying to make these decisions. So we're actually modeling how do we go forward. And the slides are not shared. Oh, well, there's really nothing going on the slides at the moment. We're modeling how do we go forward without any NIH support for a year or two or three. And because of the people on this call, that is not as unthinkable a circumstance as we had feared. And so we're incredibly thankful to all of you on that. So we've gotten asked the question, how much of your budget comes from philanthropy? And at this time, it's around 50%. And that is an enormous amount of money for a lab. It's highly unusual. It testifies to the generosity of you, your families, and the Merkel community more broadly. We've historically gotten 50% from the government. More recently, that has shifted to various foundations at the state level and cancer foundations and such as the Mark Foundation for Cancer Therapy and such. And as we're entering this inaugural year of the Merkel Cell Carcinoma Institute, that organization and all of you are even more important as we get through this unprecedented, challenging time with our government. We've used the funds that we've gotten from all of you in so many different ways to develop the circulating tumor DNA test, to develop and improve the merkelcell.org resource that's used around the world by sort of 80,000 people per year. It's amazing. So, and then one of the most impactful things and a big topic for today is our wonderful, wonderful clinical research fellows. So I want to introduce you to them. Many of you know them very well. Let's see, now I will share my screen. Okay. Let's see here, this and this. And hopefully you can see things reasonably well here. Kind of like this view. There we go. So those are our topics that I briefly mentioned. And, here's our team. There's Andy Dixon, who has been a very significant supporter of our research, and he happened to be present. Actually, he didn't happen to be present. He traveled from Florida to be present with us to celebrate his daughter Kelsey and in her honor for this institute launch. And here are fellows, Peter and Ariel. Ariel's been with us for 2 years. He's headed off to dermatology residency at University of Cincinnati. And Peter has been with us for 1 year, headed back to finish his medical school at Einstein. You guys, can you spotlight maybe Ariel first?
Yeah. Hello, everyone. Thank you again for all that you have really helped us do. And it's been such an honor to work with Team Merkel for these last 2 years. And I'm excited to take everything that I've learned here and try to help those, well, in the Midwest and see where else. But yeah, thank you for everything and the support.
Yeah, I would, echo the same things. Like, I think that, this position has just been such an honor to kind of be able to work so closely with so many different patients, be able to also do clinical research and, be able to kind of influence the future of Merkel cell carcinoma research. So it's just been an incredible honor. We wouldn't be able to do it without all of your support. So thank you again. It's bittersweet, for sure.
Super. And then let's go with Nikki next, if you're in the spotlight mode. Nikki, tell us where you're from and a brief description of what you're looking forward to. And
Yeah. Hi, everyone. I'm Nikki Boucher. I'm from Washington, and I'm a current student at Washington State University. I'm very excited and honored to join the Merkel team and really looking forward to meeting you all.
And you're a student at Washington State University in the medical school.
In the medical
and
school,
what
yes.
year?
I'm
In between.
in between my
Yeah.
third and fourth year.
So heading to last year and then taking off a full year to focus on helping us in Merkel land and so many of you. Okay, Victoria.
Hi everyone, my name is Victoria. I am from Idaho and I go to the University of Washington School of Medicine. I'm also like Nikki between my third and fourth year and taking a year off to be working here. I'm super excited to meet everyone, to get to know the team better, and to be part of some of the amazing research that's coming out of this institute.
Super. Well, I'd like you all to join me in thanking Ariel and Peter for their contributions. And it's like hundreds of messages a week, and dozens and dozens of phone calls and quite a number of in-person visits. And it's just an enormous amount of tracking data on all of these patients. It's very complex. And boy, could we not do it without this team also helping to enroll folks in research. So thank you so much, Ariel and Peter. I think you all know that philanthropic support not only enables that, it fully pays for their work. We have had, over the years, many people tell us, my dad, my brother, my wife, whatever, didn't have the kind of care that I would have wanted them to have. And I'm giving you this amount of money or support in order for you to give the kind of care, regardless of money, regardless of anybody paying. A lot of this is providing, quote, free support to people, sometimes in other countries, just gathering their data and having it get discussed at our tumor board. And our doctors giving advice to their doctors directly. And that's how we do it. So we work with their other teams. So it makes us super proud to be able to do this. And that's all because of the support that you guys have provided. Thank you so much. So I'd like to introduce you now to Geraldo Rodriguez, who's finishing up his time. I'll stop my share because, well, actually, for one more moment here, here's a picture of Geraldo at his poster at one of the recent meetings. He's a graduate student in molecular medicine and mechanisms of disease, the PhD program here, and he's co-mentored by Dr. Justin Taylor, who's a B-cell immunology expert. And Geraldo is going to, now I'll stop my share here if I can. Here we go, stop. Okay, and he's going to start his share. And Geraldo is going to tell us some exciting updates about a topic that patients are often very interested in. Geraldo?
Hi, everyone. Great being here today, and thank you, Paul, for the introduction. So today I want to talk to you guys about your gut microbiome, or essentially what's in your poop, and how what we can find in stool can actually help you at potentially fighting tumors in itself. So a little bit of background on what the microbiome is. So these are essentially bacteria that live in your large intestine, and there are a bunch of different bugs that live there. I think Paul and I were earlier discussing that might be pounds of bacteria that actually live within your large intestine, and they're actually quite beneficial for overall functioning of your body, particularly for the immune system. So your bacteria actually process a lot of the foods that you intake that you're not allowed to process on your own. And they provide this like boosting to your immune system, and they also prevent you from getting infected by bad bugs. So oftentimes you eat food, and when you get food poisoning, actually, it's because your gut microbiome was not able to fully eradicate that bad bacteria that got you sick. But eventually they come back and they kill that bad bacteria, and hopefully without the need for antibiotics. So these are the really good benefits that you get from your gut microbiome, and it's super important for health overall. But there are also associated risks that come with having bacteria living with you. So depending on the bacteria that you have, you might have a higher risk of obesity and colorectal cancer. You may also be more inclined to having inflammatory bowel diseases, especially autoimmune diseases. And if the wrong bacteria are colonizing your large intestine, you can also have opportunistic infections if you have another sort of illness. But the very interesting thing that I'm excited to talk to you about today is that there's a lot of really cool research out there that is now looking at how the microbiome is actually influencing cancer patient outcomes in skin cancers. There's virtually nothing known about how the microbiome affects Merkel, and that is essentially what we want to do. We want to investigate how the microbiome itself may be affecting MCC outcomes. But a little bit of background on what's been done in melanoma. So Jennifer Wargo is this fantastic scientist, and she's a pioneer in like studying the microbiome in melanoma, especially in folks that have been treated with cancer immunotherapy, such as anti-PD-1. And she found that when she looks at the bacterial amounts that live in your gut, so patients that have more diverse bacteria, so as evidence in this image here on the left-hand side, patients that have a variety of different bacteria, like so a lot of red bacteria, blue bacteria, different color bacteria, do significantly better. They respond better to immunotherapy outcomes.
And Geraldo, let's remind people, so each dot in the purple here under the responder is a person.
Correct.
Right?
So,
Each dot is the person, and it shows how diverse the bugs were in their gut.
Correct.
So if you're higher up in the top here, you had more diverse bugs in your gut.
exactly. Correct. And whereas if you had less diversity or like just more of the same bacteria and fewer, then patients did not respond as well. And this is something that has been validated in across different melanoma studies. And there's actually currently clinical trials that try to influence the microbiome to essentially improve immunotherapy outcomes. But the big question, so this process where the microbiome in your gut is affected or not working properly is called dysbiosis. And I think it's generally well accepted nowadays that dysbiosis or lack of bacterial diversity in your gut is actually detrimental for cancer outcomes. And the big, so how does this happen? So in your healthy gut, so this would be your large intestines, you have plenty of bacteria that live there. And it's actually the amount of bacterial diversity that you have in your gut is actually mediated by your own immune response, like your immune system. So the way that works is that you have these cells called APCs or antigen presenting cells here highlighted in purple that can sense that bacteria. They can probe the bacteria that live in your gut, and they can actually modulate how much bacteria there is by communicating with these with so-called T cells. So these APCs sense the bacteria, then they communicate with T cells, and the T cells can then communicate with these other types of cells, called B cells. And those B cells are the ones that can, upon stimulation with the APCs and the CD4 T cells, they can become what's known as IgA antibody secreting B cells. So these IgA molecules, these are antibodies that are specialized at being secreted out of your intestinal wall into your large intestine, and they can actually specifically bind to bugs in your intestine. And through this mechanism, we think that the antibodies that are being secreted can tell the immune system, okay, we have too much of this bug, but we don't have enough of this bug. So essentially, they normalize the population of bacteria that live in your gut. And this is how your immune system can actually modulate that bacterial diversity.
So I want to say that again because some of those words you don't need to you don't need to understand all of those complicated words, but the immune system has a way to regulate this diversity of the bugs in our gut. And we don't fully understand it yet. We don't understand how much it may or may not be playing a role in Merkel cell carcinoma patients. And a lot of this has been done in mouse, right, Geraldo? So it hasn't so much yet been done in humans. And as a side note, we thank you for your poop.
Yes.
It's actually harder to get people's poop than it is to get their blood. They're like, no problem, I'll give you some blood. But no, nothing from the bathroom from me. So we appreciate when we ask our patients and you're willing to do the bathroom deed. It turns out it's important, and our bacteria inside the poop actually have a variable amount of this antibody stuck on them in a very specific way. And so that's what Geraldo's planning to study. Go ahead.
Thanks, Paul. Okay, so, and this is what happens essentially in a healthy gut. There's this perfect harmony between your immune system and the bacteria that live in your intestines. However, if your immune system is not behaving how it's supposed to be, such as in the case of having an immunosuppressive state, or like your immune system is downregulated, or if you have an inflamed gut where your immune system is like too active, then you start disrupting this perfect harmony between the bacteria and the immune response. So in the cases where there are not enough immune cells regulating the microbiome, as evidence on the left-hand side, you have all these bacteria that are overpopulated, and one tends to take over all the other ones, and that is really bad for a variety of reasons. And in the case of an inflamed gut, you have too many of those immune cells killing the good bacteria. And you also start generating other kinds of responses that you can detect in the blood, and that's usually not good. So as Paul was alluding, the big question that we have is, how are these different responses affecting MCC patients? And we're very convinced that we're going to find something interesting in this, and I hope that you can appreciate that from my not very patient-friendly slides. So thank you, Paul.
That's all right. We can translate. That's okay.
For being my translator. In addition to that, we also believe that there's something that you can actually start doing right now, independently of whether you're a patient affected with MCC or just to live a healthier lifestyle. So it turns out that fiber is really, really, really important for your diet, especially for the bacteria that live in your gut. So in the large intestine, you're actually, your large intestine should be covered with this nice cushy layer of mucus that depends a lot on how much fiber you eat. So if you eat a lot of fats and not enough fiber, that cushy layer essentially becomes depleted. And when that cushy layer becomes depleted, you can actually have tears in your intestine that allow the bacteria that is not supposed to go into your blood to enter the blood circulation. And we call that a leaky gut. So when that happens, that can lead to a lot of different problems. And we think that we can detect that in the blood. And we also believe that these responses that we can detect in the blood, if we see them, may actually be associated with bad MCC outcomes. So the moral of the story here is like eat a lot of fiber. Diverse fiber is really good. And we also want to investigate this within the big aims of our project in the future directions. So this is essentially our goal. We want to know how it is that the gut microbiome and your gut immunity affects MCC outcomes. And as Paul mentioned, and I have been reinforcing, it's a big unknown right now in our field. We virtually have no clue what is happening because no one's looked. So hopefully we can start looking very soon. And matter of fact, we actually already have a lot of stool samples from patients. So thanks so much. We also want non-MCC controls as well. So if you feel inclined to, please email us and we'll send you the paperwork and you can send us your poop. We'll definitely look at it. So, and this is just sort of like what we plan on doing. We have two big aims. One of them is just literally take the stool samples that we get from patients as well as non-MCC controls and start looking at what kind of bacteria live within those stool samples. We do that by essentially looking at the different DNA that is contained in the poop, and we can actually identify the different bacteria that you have in your poop based on the DNA sequences that we see. So that would be the first thing. Just look at MCC bacteria, look at the bacterial DNA, and then start correlating the kind of bacteria that live in MCC patients to outcomes. In addition to that, we also want to use this fancy technique that allows us to see which bacteria are being seen by the immune system. This technique is called microbial flow cytometry, but it essentially allows us to know which bacteria are actively being recognized by the immune system and separate the bacteria that is recognized by the immune system from the one that is not recognized by the immune system. Yeah, I think Paul wanted to translate. Okay, that worked?
That's great. Good.
Excellent. And so we're very excited to also say that this, as Paul has been mentioning and everyone else, MCC is a big team sport. And I'm highlighting this random PDF file that I will explain what it means. So the first time I ever met Paul, about 5 years ago, I was actually interviewing for grad school here at the University of Washington. I was walking into his office. It was a rainy day in Seattle. I fell. I was wearing a black suit. It got super muddy. And
I was like, who is this bozo with mud all over this very silly black suit?
I had never heard of Paul before, and I was like, oh my God, like now I'm gonna meet with him. He's gonna determine whether I can go to grad school or not. And then the first thing he looks at me, he's like, are you okay? And I'm like, yeah, sure. Like my shoulder was not feeling well. But we were supposed to meet for 20 minutes, and we ended up talking for like more than an hour. Because he's such a fun dude. And like and at the end of the whole meeting, he prints out this article that he wrote a few years ago, where he essentially talked about how research is a team sport. And we went through it, and then he wrote that little note and stapled his business card to it. And 5 years later, here I am talking to you about that story. But I think it's really important to highlight that because this fall work that we're planning on doing is a collaborative effort between 3 big labs at the University of Virginia, here at the University of Washington, and the Fred Hutch. And this is thankfully was recently awarded, like actually yesterday, we found out that we got $150,000 from the Bezos Foundation to start asking these questions and doing all the sequencing. This will be in collaboration with Justin Taylor, who's my current co-mentor, as well as Dr. Megan Koch, who will be a new addition to the MCC family. And she does fantastic work in gut immunity, so I think we're going to have a lot of fun, the 3 of us working together, and me sort of leading the way, I guess. Paul's going to take a back seat on this one, I think. But
more bad poop puns.
But a few years ago, I had this fantastic mentor that would always tell me to find a mentor that believes in you, even when you don't believe in yourself. And in science, like there are so many different times where you really don't believe in yourself because the science is not going well. So I think Paul and Justin and Megan are fantastic at doing that. And I try to pay it back. I've been mentoring some students of my own. I love working with undergrads. They have a lot of enthusiasm and they keep you young. And energetic, and they have all these like silly sayings from Gen Zers and millennials that I'm also learning about, and we're also teaching Paul. And 2 of them are actually here on this call, and I want to highlight them.
Next slide, maybe. Yeah, there you go.
One of them is Claire Park. Claire.
Yeah. Hi.
Yep, you're pinned.
Okay.
Yep.
Great. Hello. So my name is Claire Park, and I'm a 3rd-year undergraduate student at the University of Washington. I'm currently studying biochemistry and Korean with the goal of being a doctor in the future. It's been a little over a year since I first joined the Nghiem lab, and I'm really excited to get started on this project. My role in it will be mostly doing bench work and clinical data analysis. And from this project, I'm interested in both improving my skills as a researcher and also learning more about adaptive immunity and its relationship with cancer outcomes. Yeah, and then outside of research, I also play the violin, and I like sewing and petting my dog. Nice to meet everyone.
Scotia.
Hi everyone, I'm Scotia. I am also a 3rd-year undergraduate student at the University of Washington. I'm studying molecular, cellular, and developmental biology. I'm from Sammamish, but also from Toronto, and I hope to pursue an MD PhD in the future. And for this project alongside Claire. I'll be doing a lot of the bench work. So all the poop samples that are sent then end up on our desks, and then we handle them. And I'm really interested to see the potential applications for this project as a biomarker and the translational side, because this project is really focused from going bench to bedside. So I'm very interested in that translational approach for this project. And when I am not here in the lab, you can either catch me playing violin. If you can't see, Claire and I have played with Dr. Nghiem and quartet. That's us actually in the top right, practicing at his house with his dog. And you can either come me rock climbing, sewing, or reading.
Hello, I'm sorry I'm late.
I will
This
stop
is Mike
sharing.
Rainbow. I'll mute.
So that is us, Team Poop.
Well, we're so proud. Let me clarify something for just a second about Geraldo. That he is talking about a project he's just starting now. He has done a beautiful job doing B-cell biology, which is just about to be submitted as a very exciting paper, and that may have implications for treatment in the future. It definitely helps us to understand how the immune system fights the cancer, but we chose not to go into that today. It's even more complex, and we will fill you in on that later, especially if it's starts moving towards people. So I will once again share my screen here, and I did want to tell you about a few words before we go to questions very soon. A few words about our wonderful philanthropy team. So Sherry Dotson and Megan Little are here today. Thank you. And they're really helping to plan all of these details. Thank you guys so much. And it's just, again, this week, as we've said, and I haven't slept that well, that we don't have any NIH funding for the foreseeable years. I do not know what's going to happen. It's just incredible that we have this philanthropic support, and it's made us have to not make horrible decisions about not doing the things that we're excited about. And we're really thankful to them. Do you guys want to say a word at all about
Sure.
Yeah.
I'm not sure if I'm being spotlit, but hi, Megan Little and Sherry to my left. We've had the pleasure of partnering with Team Merkel for some time now through Fred Hutch Cancer Center Philanthropy, and it has been a delight to work with all of the folks who presented today in partnership, and to build relationships and learn stories and receive support from many of you on today's call, and many who will be viewing this at a later date. So thank you for all that you do to help advance the work here in the lab as well as the Institute. And we invite future conversations if you're curious to learn more about how you can support us. Anything that you want to add, Sherry?
I don't think so. I'll just add a thank you so much for being here today and for your incredible support. We really appreciate it.
Thank you guys so much. We could not do it without you. Absolutely. Let's see. So we just want to tell you that the Merkel celebration will be on Monday, September 15th, a tiny bit later this year than it has been. That's partly so that many of us can go to a big international Merkel meeting in Stockholm shortly before that. But we look forward to seeing many of you during that. And I know that Mike Rainbolt just joined a couple moments ago and said a super quick hi then. But thanks to the generosity of them, all of our donations and his wife, Burma, all donations to Team Merkel, which is, we're calling ourselves Team Skin in the Game, for our Obliteride will now be matched dollar for dollar up to an additional $20,000. So thank you so much, Mike and Burma. And so with that, I think we'll go to Q&A. And I think there were already several in the chat that I was kind of liking because I saw them come up and they were really great questions. I'll tackle one. How is this related to taking probiotics in terms of the gut? And there is, you're exactly right, Jamie. I seem to remember an article suggesting that probiotics were not helpful and it's not in Merkel, but it was in melanoma. And we're kind of extending the melanoma data, wondering if it might be the same for Merkel. I don't think it's a giant guess or extrapolation. It's also a cancer that's very visible to the immune system. So those data, you're exactly right. If you take probiotics, so that's like living bugs. They only live in you for a very short amount of time, and they don't fundamentally really change what's in your gut. And the surprising thing there in some of those studies was there was a suggestion that taking quote ideal probiotics does lower your chance of having a good immune response against the cancer. So in that case, we're talking about people with advanced disease who are either taking, one food source or another, and the probiotics did not seem to be helpful, maybe detrimental. So we wouldn't encourage that. What seems to be better, and then this gets on to Shell Kappan's question, does soluble versus insoluble fiber make a difference at all? And maybe Geraldo and team can comment more sophisticatedly on that. I think the high level is a diverse source of fiber is the best, which would be a mixture of soluble versus insoluble. And I think that, the implication there is, maybe we can just take a ton of Metamucil and everything's going to be fixed. And I do not think that that's the case. Personally, I take some Metamucil. I have for a long time. It's probably good for our health. That's what I got convinced of in medical school. But it's not alone enough. And really, you want diversity of those fibers. And we can make sure that we send out to this team. We just were working yesterday on a handout of, all the different food sources, like black beans have a ton of fiber and artichokes have a ton of fiber and, whole grains have a lot of fiber and whole fruits are really good. So you want to have a good mixture. And that's what's associated with supporting diverse bugs, because that's their only source of food. You eat sugar and that's all absorbed in our body and probably not good for us in excess for sure. But that's not food for the bugs. The food for the bugs is the stuff that our bodies can't digest. Okay. While study going on, a healthy gut is good regardless. Would you put a healthy gut regimen in place for new Merkel patients? 100%, Larry. We already are encouraging that. We're going to put more data out there. We think that for cancer reasons, for general health reasons, for every reason, a diverse source of foods that are higher in fiber is really, really good for health. So it's not premature, as is exercise. And, you don't have to go crazy with exercise, but find something you like to do, at least 20 minutes of walking or gardening or certainly aerobic exercise. Really good for your immune system and the ability to fight cancer and everything else in life. So I think diverse food sources and exercise are really good. So Richard, note posted in Merkel group recently. Oh, yes. Pyravinium pamoate is being effective against Merkel. I think that those data work in cell lines and maybe in some mice, but man, oh man, do we not know anything about whether in humans. I would not do anything radical about that at all at this point. And then Krista put in a link to our team on Obliteride in case you are motivated by that. And we definitely want to thank the Rainbolts. And oh yeah, Krista reminds me we are adding a lifestyle page to the merkelcell.org website. So that will include optimal food sources and information about how to optimize your diet, as well as the importance of exercise. Barb, how does immune therapy like Opdivo affect gut health? So the other way around. I don't know too much about that. What I can say is antibiotics affect your gut health a lot. And taking antibiotics right before you start immune therapy decreases the efficacy of the immune therapy. So it kills a bunch of good guys. It changes things around. I mean, if you got a big infection, you got to take antibiotics, when it's the right thing. But antibiotics suppress the diversity. I'm not aware of the Opdivo types of drugs changing meaningfully gut health typically. Stuart, recovering from immunotherapy-induced colitis. Okay, ugh, not sure how to best rebalance my microbiome. Right. Well, I do think at the very beginning, just some old-fashioned yogurt with living cultures is a good start. That's the best data for living bugs to help reconstitute. But you want to, as soon as your gut's feeling kind of okay, start getting to diverse fiber sources, because that will really change and increase the diversity and health of the gut microbiome as quickly as possible. Those are the questions we have. Put more questions in. Any thoughts on TVAC for Merkel treatment? Oh, yeah. So TVAC is Telimagene laherparepvic, and that is a modified herpes simplex virus. So the cold sore virus that's been modified to take out some dangerous parts and has had some immune stimulating parts put in. And when it enters a cancer cell, it will kill that cancer cell, this modified version. And it will also, grow a little bit inside that cancer cell, but it can't grow outside the cancer cell. TVAC is the first generation drug. It's a little over 10 years old. It is injected directly into skin lesions or maybe injectable lymph nodes that you can feel. And it causes an immune response against the cancer, and it kills cancer cells. It works really well. Now, if you've got a whole bunch of disease all over the place, that's not for you. You need immune therapy. But if you only got one or two things, and especially if you can't take or aren't benefiting from standard immune therapy anymore, TVAC or those derivatives work most of the time. They will shrink and or eliminate the lesions into which they are injected, and they're pretty darn safe. So it's a very good tool. We use it in those settings that I just mentioned, like if we don't have a bunch of other good thoughts or treatments. Yeah, Michelle, great. You can just put your note in there. So I will go back to this big issue of funding and the National Institutes of Health. We've never had, in the history of the NIH, there's never been a 30+% cut. And I really hope that doesn't happen. Cuts have been called for in the past, but they haven't really gone through. And that's what I'm hoping and praying will happen by September of this year, when our current budget ends and a new budget has to be adopted for our country. I do think that your statements sent to your representatives can make a difference. And especially, honestly, if you are represented by a Republican in the Senate or the House, those are the people that can affect things. All the Democrats don't want to see this cut already. But some Republicans don't, but they're kind of following the rules. And I'm not in a lobbying mode here. That's not what I'm supposed to do. I'm kind of telling you facts. A 30% cut will mean something that's never happened before. We just don't know what's going to happen. They're literally going to have to take away grants that were given to people before. And new grants probably can't happen at all. So I hope that doesn't happen. And I hope that you all believe that research like we've seen in this field has changed lives. I know I can see a lot of the people on this call today, honestly, they wouldn't be here if it weren't for the progress that we've made. And conversely, I know of a lot of people that would be on this call if we had made more progress. We actually know what the mechanisms are in many cases that the cancer is using to escape our current immune therapies. They downregulate class 1 MHC. They, block the immune cells from getting into the cancer. The class 1 MHC is necessary for the immune system to see the cancer. They hide from natural killer cells. We have ways to treat all of those things in theory and in mice and in cells. And we have trials and things that we want to move forward with and can't. The clearest example is our transgenic T cell receptor trial. We have $1.2 million from the Washington State Foundation that we can't use because we don't have matching funds for it. So from the federal government. So things are on hold and it's hard. And, I do think that Congress people listen. And if you, it's one thing for scientists to say, hey, my job is at stake. They kind of yeah, we kind of know you're kind of valuable, maybe. But if a patient or a patient's family member says, we know the NIH has done amazing research that has led to all of these things like the new drugs that control weight and diabetes. And, so many things for heart disease and so many things for cancer. It's all based on NIH results that then eventually find their way to pharmaceutical companies. So anyway, the matching fund amount is $1.2 million that we were going to use from the NIH. That would have covered it for that. And we're not sure how we unlock then those state funds. So anyway, those are my thoughts. I don't and I hope that we as a country and are, without a doubt, the United States is the strongest biomedical research entity in the world. And we literally have people who are retiring early, who are leaving research, and who are going to other countries because our funding is cut and long-term threatened. So this is our world leadership to lose, and I hope it doesn't happen. I remain optimistic that things will change and the NIH budget will not have a 37% cut. The 5 Calls app makes calling senators very easy. Okay, there's a link. Okay, thank you. Sorry. Yep. There are a number of ways to do that. You can just Google too. How do I easily contact? And I know that our patient representatives, Larry, Stewart, you guys have sent out things through the patient support group, which we're not allowed to do outright, direct any lobbying or anything. I can tell you facts. I can tell you if that cut happens, we don't know what's, really bad things are going to happen to research. Yeah.
There's one more question from Ed.
Ed, read it, Krista.
Oh, sorry. Is the matching?
Oh, I addressed that.
Oh, you did. Okay.
Yeah. So again, we thank you all so much. Maybe turn off my spotlight so I can see other people. Thank you for joining. We are excited about this Stockholm meeting. We'll have a very good contingent at the International Merkel Cell Carcinoma research meeting at the end of the summer. We'll be telling them about trials to promote and explore single fraction radiation that are going to change the way radiation is given around the world. We're excited about a circulating tumor DNA trial where some of you know this story, if you have evidence in your blood that the cancer is coming back, but scans can't find it yet, we're going to start a trial, we hope, if we can get the funding. Again, that's another federal funding source that we were hoping to use to test whether immune therapy makes sense. Again, when we can see it in the blood, but we can't find it yet on a scan. That's an exciting direction that we want to go. What am I missing, team?
Thank you to Stuart Landers. So the grant that we just got actually needed patient advocacy, and Stuart was very kind to support the efforts for us to get this money. So thank you so much for writing a letter for us and helping us get the startup funds to start looking at poop.
Thank you, thank you. Anything else, team? We did it. Any other questions? You can send us emails afterwards. And I just, once again, personally, in this very, very, very hard time, just can't tell you how meaningful it is to have your emotional, logistical, financial, blood, poop, sweat, tears, whatever support. It makes all the difference in what we're able to do, and literally amazing to be looking out on people that we know wouldn't be there if it weren't for the efforts of this group. Thank you. And I mean the big group, the big team that definitely includes and is led by all of you who give us our ideas about what research we need to do. Thank you.
Thank you.